Hi everyone. In Lecture 9 recording, we are going to delve into drug stability and incompatibility. But bear in mind, in this recording, it's only meant to give you an introduction of the main concepts. As you move along the program, when you have more exposure to different dosage forms, you will actually learn more about the different drug stabilities and incompatibilities associated with the specific types of dosage form. So these are the learning outcomes for this lecture. You should be able to define drug stability, list the different types of drug instabilities, explain the main mechanisms of drug degradations, therefore lead to drug instabilities, list the factors that may affect drug degradations, define incompatibilities, and explain the different types of incompatibilities. So basically, this lecture recording will be principally consist of two main parts. In first part of the lecture, I'll focus more on discussing drug stability. In second part of the lecture, I will discuss more in terms of the drug incompatibilities. So what does drug stability means? The proper definition of drug stability is the capability of a particular formulation in a specific container or closure system and remain within its chemical, physical, microbiological, therapeutic, and toxicological specifications at a defined storage conditions. So putting things to be simple and more understanding is that for each drug that should be to be marketed, they must fulfill 5 types of stability, okay, which are the chemical, physical, microbiological, therapeutic, and also toxicological stability. So let's look into slightly more detail in terms of these 5 types of stability. So in terms of chemical, it means that for each drug, the active ingredient should remain its chemical integrity and label potency within the specified limits of time. So within that time frame, okay, let's say 2 years, so the drug should not change in terms of the chemical entity, and also it must remain sufficiently concentrations whereby it should achieve the required effects. For example, if it is for a treatment, it must achieve the required treatment purposes. Okay. In terms of physical, it should actually has the original physical properties, include the overall appearance, the palatability, meaning in terms of the taste or the smell, uniformity, dissolutions, and also suspendability are retained. So in a case, if it is a tablets or solid preparations like capsules, then the dissolutions properties is very much of concern because it actually affect the bioavailability of the drugs, meaning the drugs, the solid drug need to dissolve, start releasing its solids, and then dissolve, transfer its solid mass into a solvent or dissolving medium, and then absorbed. So the dissolution is actually affecting the bioavailability of the drug. Suspendability will be much applicable if the preparations or the dosage form is a suspension. Okay, so the overall physical stabilities include all those that I have mentioned just now. Now, in terms of microbiological stability, it means that the sterility of the product or the resistance to microbial growth, okay, within the specified requirements. If there is any case that the drugs or the dosage form consists of antimicrobial agents, then the effectiveness of the antimicrobial agents must be maintained during the specific time frame. So for therapeutic stability means that the therapeutic effect of the drug should not be any changes. And finally, there should not be any increase in terms of the toxicity of the drug, meaning there should not be any generation of toxic substances in the drug itself. So the common types of drug stability instabilities may actually demonstrate as, for example, a drug or excipient degradations that happens via the chemical, physical, or biological mechanisms. Okay, so in this slide, I would like to show you some of the common types of drug instabilities. So the first example would be, for example, the loss of active drugs. Now, this can be happen, for example, the drugs undergo hydrolysis reactions, such as aspirin undergo hydrolysis, or oxidation of adrenaline. So this actually cause the drug become inactive form. Okay, they no longer carry the therapeutic effect. The loss of vehicle from the drug or the dosage form. For example, evaporation of alcohol if it is alcoholic mixture, evaporations of water from an oil in water creams, the loss of content uniformity. If you look at the examples of picture here, so the examples of this bottle is actually consists of contains of emulsion. An ideal or proper emulsion, you should not see any separations of layer over here. So over here, this is a drug instability whereby the emulsion already has creaming, okay, on top of the layers. You will see a layer of oil separated out. This is called creaming of emulsion. And if it is the case of suspension, usually suspensions, we can actually easily redistribute the suspension by shaking. Okay, so the particles will start redistribute themselves. However, there are situations whereby the particles just sediment and they form a very impact solid formations and which are not easily distributed back. So in this case, it may actually cause content uniformity is not uniform. The drug is no longer uniform. Another type of drug stability that can happen could be that reductions in the bioavailability. As I mentioned earlier on, this actually is relevant to the dissolutions of the usually the tablets or the solid dosage form of capsules, for example. And that may happen, for example, due to aging of the tablets. Aging means the tablets has been have been kept for a long time and it become old already. So this actually may change how the drug dissolve. The loss of pharmaceutical elegance. Okay, for example, there's fading of the colored solutions or fading in terms of the tablets. As you can see on the picture here, some of the tablets actually the color already changed. Productions of toxic materials. Okay, this could happen probably due to the breakdown of products or the degradations of the product, and it create toxic substances during its degradation process. And these toxic substances actually cause hazardous effect to the patients. So how the drug instabilities can happen? Okay, so there are three main type of mechanisms of drug degradation, namely the chemical degradation, examples like solvolysis or hydrolysis, oxidation, photolysis, polymerizations or optical isomerization. So we will look into each type of this mechanism in a short while. The physical degradation such as polymorphism, vaporization, water loss, absorptions of water or particle sedimentation, which we have talked a bit just now, yeah? And finally, it could be due to biological degradations, mainly due to the contamination and spoilage by the microorganisms. Okay, so in terms of chemical degradation, the most common chemical degradation is actually hydrolysis or solvolysis, right? So this basically means the drug degraded or the excipients of the drug degraded when they react with solvents. And the most commonly used solvents in our field is actually water. Okay, so to put things into much more easily understand, is that there's actually breaking of the drug molecular bond when reacting with water. Okay, so the common drugs that are susceptible to hydrolysis are those drug compounds or any compounds that carry the carbonyl compounds. Okay, for example, esters, amides, lactones, and lactames. So drugs that carry esters compound, for example, like aspirin. Okay, drugs, example of drug that carry amides compounds such as sulfonamides. Okay, spironolactone carry lactones in their drug structure. Penicillin carries a beta-lactam ring in their drug structures. So all these chemical groups or chemical compounds are highly susceptible to hydrolysis because the carbonyl carbon is actually an electron deficit compound whereby they are easily attacked by the oxygens of the water molecules. Okay, so by undergoing hydrolysis, for example, esters, hydrolysis of ester, it can actually break down into carboxylic acid and therefore alcohol. If it is amides, then it will break down into carboxylic acid and amine salts. In terms of oxidation, basically it means there's actually an increase in the number of carbon-oxygen bonds in the molecule, meaning there's additions of oxygen, or a reduction in the number of carbon-hydrogen bonds, meaning a removal of hydrogen. So oxidations usually involve the atmospheric oxygen. And if the oxidation process that involves atmospheric oxygens, it is known as auto-oxidation, and it is a chain reactions process whereby it usually involves 3 stages. So in the initiation phase of the auto-oxidation, it results in a formation of low concentration of free radicals. And this process, the initiation process, is actually highly promoted by light or heavy metals, for example, copper or iron, that commonly found in the pharmaceutical product as a contaminants. During the propagation phase, the concentrations of free radicals basically increases a lot. And this actually even speed up the degradation process, whereby with the presence of oxygens in place, the oxygens together with the free radicals, they lead to formations of hyperperoxides. And these hyperperoxides basically form very stable oxidation products. In the termination phase, basically oxygen and also drug has been used up. Therefore, the rate of the reactions actually reduce. And at this point, all the free radicals already combine with the breakdown of the product and form very unreactive end products. So common drugs that undergo oxidation such as the riboflavin or vitamin B2, ascorbic acid or vitamin C, adrenaline, iron salts or ferrous salt, and also vegetable oils. Okay, all these drugs or all these materials, agents, can actually susceptible to oxidation. And oxidation is usually demonstrated as a change in the color. For example, take adrenaline solutions. It may actually cause, after oxidation, it may actually change from a clear color solutions, there's no color on the of the solutions, and it become red colored solutions. Okay, during formulations, oxidation may be inhibited when we add antioxidants such as, for example, sodium metasulfate, sodium sulfate. So depends on the dosage form again in upcoming future when you are exposed to different types of dosage form, and you may come across different types of antioxidants being used in different dosage forms. So photolysis is basically the degradation of drugs or the drug excipients that caused by light. Okay, so the photolytic reactions, meaning the energy from the light radiations, the light can be from either the room light or the sunlight, and it absorbed by the molecules. So this actually create energy, okay, within the molecules, okay, and then it will cause the bonds from dissociating. Okay, so meaning it break down the bonds. All right. So during the photolysis reactions, common terms that you may come across include, for example, photolabile. Photolabile means light sensitive drugs. Okay, drugs that are light sensitive, we call them photolabile. Photosensitizer, on the other hand, it means that the molecules absorb light, but they don't cause any reaction. They pass the energy to another molecules, and then it subsequently causes degradation. So we call that drug or agent photosensitizer. It only absorbs the light energy, the energy from the light. Okay, so the UV light of the 300 to 400 nanometer wavelengths are usually the most damaging wavelength that in terms of drug degradations. So most of the photolysis reaction actually involve oxidation mechanism, although other mechanisms also may be in place. Okay, examples of drugs that may undergo photolysis include, for example, nifedipine, okay, riboflavin, chromazine, ciprofloxacin, and vitamin K. So usually those drugs that undergo oxidations, the color may actually turn either yellow or it become darkened. Okay. For drugs, okay, to protect from photolysis, usually we can change in terms of the packaging. Okay. We make sure that we use light protected packaging like strip pack, okay, strip packaging, or amber bottles, basically to prevent or override the photolysis reaction. Polymerization is another type of chemical degradation. Okay, it basically happens when the reactions of a drug molecule with another molecule of the same drug, and it results in the formations of a very complex or larger molecule, usually called a dimer or polymer. The polymerization's degradations mechanism is less commonly known in the pharmaceutical field. Okay, so two common examples will be, for example, amoxicillin. Apart from hydrolysis reactions at the beta ring, the polymerizations can also affect the beta ring, okay, whereby there's nucleophilic attack of the beta ring by a amino group. Okay, another examples of polymerizations that happen in a disinfectant known as glutaldehyde. Okay, so the degradation of glutaldehyde when it's at the more alkaline pH, so the degradation process is known as polymerization. Okay. Lastly, optical isomerization. This is a process where there's a change in terms of the optical activity of the drug, and this actually affects the biological or therapeutic activity of the drug. The most common isomerization is known as racemization. Okay, so racemization basically it's a process whereby the optically active drug change into its enantiomer. So the more layman term is the mirror image, right, of the optical drug, and usually the mirror image has less therapeutic activity. Okay, because it could be because of the alignment of the drug structure, so it actually compromise the binding activity of the molecule with the, for example, drug receptors. Okay, so this actually reduce the therapeutic effect of the drug. Okay, so examples of the isomerization are provided, like, for example, conversions of the negative form of hyocyanamine into a racemic mixture of positive and negative hyocyanamine. So these racemic mixtures could be that the therapeutic effect is not as good as the originally optically active drug. So now let's move into physical degradations. So the common examples of physical degradations, one of it can be polymorphism. So if you have been exposed with polymorphism, so polymorphs are actually different crystals of the same compound. Okay, because of these different crystals, the polymorphs actually differ in terms of their energy, the solubilities, and also melting points. And all this actually would affect the stability of pharmaceutical dosage forms. For example, the beta form of theobromine oil or cocoa butter is actually very suitable and stable as a suppository base. But if we accidentally heating the beta form of the theobromine oil too much, it actually convert into its alpha form of polymorph. So the alpha form is actually not stable as compared to the beta form as suppository base. The next type of mechanism of physical degradation is actually vaporizations. Okay, so this is particularly applied to drugs which are volatile. Okay, so the drug basically lost through vaporization, and it can even happen if the closures of the container is not well fitting. So drug evaporate vaporize from the container. Okay, so the common examples of drugs that actually degrade through vaporizations like glyceryl trinitrate, alcohols, camphor, menthol, and chloroform. So these type of drugs, we make sure that they are packed with airtight container. Okay, airtight container, well closed and airtight container. For glyceryl trinitrate, for example, usually if we supply GTN, glyceryl trinitrate, to patients, we will supply as original bottle to the patients directly, the bottle that comes directly from the manufacturer, okay, which is well closed and airtight. Right. So another type of physical degradation will be water loss. This can happen in any kind of dosage forms or pharmaceutical preparations that contain, of course, water. So for example, the evaporations of water from liquid preparations, this can actually increase the concentration of drugs and therefore increase the possibility of crystallization of the drug, because the concentration is too high, then the drug crystallizes out. So the water loss from oil in water cream, another examples, it can actually reduce the overall volume of the preparation. And when the water loss from the cream, it actually produces a very rubbery feel on the surface of the cream. So this actually changed the appearance of the cream. And eventually, if the water loss is too much, then cream actually crack. Crack means it's separated in two layers, for example. Absorptions of water is another type of physical degradation. For example, those deliquescent compounds, for example, calcium chloride and potassium citrate, they absorb water and they will dissolve or become liquid after absorbing the moisture from the atmosphere. Okay, hygroscopic compounds, for example, glycerol, they only absorb moisture. However, they will not turn into liquid or they won't dissolve. Particle sedimentation, for example, a good example is creaming of emulsion, which I mentioned earlier on as well. Creaming of emulsion is basically the aggregations of the globules, oil globules. Bear in mind, emulsion is a dosage form that contains oil and water within the formulation. So if emulsion which is not stable, it may cream, and you will see that either a layer of oils separated out and floating on the top of the emulsions. So these are basically the globules. They aggregate together and they merge among each other and form a layer. So the creaming can be upwards or downwards, depends on the preparations of the emulsion. Okay, so lastly, biological degradations. These are a summary of the overall biological degradations. More information will be available when you go into the microbiological lecture, and also when you learn more about the sterile pharmaceuticals. Okay, so all medicines are actually susceptible or vulnerable to contaminations and spoilage by microorganisms, particularly, of course, those contain water in the formulations. So contamination means that the entry of microorganism into the product, while spoilage means that the product damaged by the microbial growth. So microorganism itself should be actually ideally excluded from all kinds of medicines, because contamination from microorganism itself can actually may cause infections, and it will be very dangerous, particularly for those patients who are immunocompromised, meaning their immune system is not well functioning. Okay, may degrade the active pharmaceutical ingredient. Okay, it probably cause any of the previously chemical degradations, mechanism of chemical degradations that we have gone through. Okay, so it also reduces products' physical stability. So microbial contamination can come from any one of the three resources, and the most commonly is actually from the raw materials, and particularly water. It may also come from the manufacturing environment. If it is a dirty environment, of course, the contamination will be high, and also personnel. If our hands are not properly washed. Personnel who are formulating or manufacturing involved in the manufacturing is not protected with protected or not wearing any personal protective equipment PPE. So contaminations like particles, our microorganism on the skin can actually shake or drop into the pharmaceutical products that is being manufactured. So usually anhydrous medicines, meaning medicines that do not contain water, are less likely to microbial spoilage because the absence of water, so they don't actually promote microbial growth that much. Okay, in most of the pharmaceutical products that contain water, usually preservatives will be necessary and need to be added. Okay, and also for multi-dose sterile products, preservative need to be in place, right? Basically to prevent the microbial growth in the product. So having go through the different types of degradations, there are actually several factors that we may consider that, and these factors can actually influence the overall drug degradations. So one of it is actually temperature. Generally speaking, the higher temperature, the higher rate of degradation reactions. Okay. For instance, every 10 degree increase in storage temperatures, the rate of the reactions may actually doubles. Therefore, it's very important that for the drug products, the storage conditions, the storage temperature must be clearly stated. The type of solvents added into the formulations can actually affect the or influence the degradation of drug, because these solvents can actually change the pKa or the acid dissociations constant, the surface tension, and also viscosity of the formulations. So different solvents may actually affect or may change or may influence a drug degradation process. The moisture from the atmosphere can also affect the degradation process. As we have gone through earlier on just now, some of drugs they may absorb moisture from the atmosphere, and it is particularly true for dry drug formulations. For example, the gelatin capsules, when they are exposed to atmospheric moisture, they become soft and then they swell. Some of the drugs may undergo hydrolysis, for example, aspirin. Okay, they may undergo hydrolysis. And presence of moisture actually promote or increase microbial growth. Therefore, it can actually spoil the drugs through the biological degradation process. So, apart from that, by adding several additives or adjuvants or excipients into formulations, we can actually influence, okay, or delay or prevent drug degradations. For example, by adding buffer salts, we can actually adjust or stabilize a pH of a formulation so that the stability of the drug can be better, can be improved. Surfactants, on the other hand, added into an emulsions or creams, it may increase, it may decrease, or it may cause the degradation process unchanged, depending on whether we are adding the correct surfactants or not. Okay, and another examples, like for example, antioxidants can prevent or reduce oxidation. And another good examples, like for example, the preservatives added into the drug formulations can actually prevent biological degradations. So it depends on different type of dosage form. Different additives or excipients will be needed to be added into formulations to increase the drug stability, thereby prevent degradation. Okay, so before we move into drug incompatibilities, a final note in terms of drug storage. Okay, I would like to highlight that all drugs have a limited shelf life, meaning they cannot be stored forever. Okay, so on top of that, if the patient's having incorrect handling or improper storage, all this can actually contribute or leads to drug degradation. Therefore, the shelf life of the drug will be shortened. It is very important, therefore, that Appropriate storage conditions, expiry date, or beyond use date should be clearly stated on all the drug labels or packaging of the drug formulations or drug products. Okay, so this table over here basically summarize or compare sort of the terms that I've brought up earlier on. For instance, shelf life. Okay, so when we talk about shelf life, it is the term that commonly used by pharmacopoeias or regulatory authorities to show or to delineate, state the period during which a drug product, if we follow the manufacturing process, the manufacturer, it should remain suitably fit. Okay, for instance, usually the common criteria is that it should be more than 90% of its potency, okay, before it actually being used or before it reach the patients, right? So usually the shelf life of all the drugs, most of the drugs is usually between 2 to 3 years. The shelf life is usually being calculated and predicted using some calculation and based on the drug stability test by the manufacturer, the drug manufacturer or the company, drug company. They will estimate, okay, the drug will be stable for example, 2 to 3 years. The expiry date is the date whereby it is commonly stated on the container or labels of the drug product. Okay, and it is the time whereby a particular batch of the product, okay, which is formulated and it should be expected to remain, okay, the same within the approved shelf life specifications, okay, if the drug is being stored as recommended conditions, okay. So basically the expiry date is the commonly date that we seen on any of the drug packaging. Beyond use date is basically the drug that is the date that is used after a preparations or a drug is being compounded. Okay, this is commonly used in the extemporaneous dispensing process. Okay, so the beyond use date is usually determined from the date that the drug is being compounded. So on the drug label, we don't really state like beyond use date. Okay, what we state is that we will state very clearly, discard after, then we will put a specific date for the patient, or do not use after and then followed by a specific date for the patients. Okay, so this table basically sort of compare the terms used in terms of drug storage or the expiry dates. So in the second part of the drug, I would like to provide an overview of drug incompatibilities. So what is incompatibilities? So incompatibility basically is an interactions between two or more, usually antagonistic substances, meaning they are counteracting each other. And it can lead to the change in terms of the chemical, physical, therapeutic properties of the dosage form. So put things in simple, it means that the pharmaceutical substance A cannot exist at the same time as pharmaceutical substance B. So this means incompatibilities. So incompatibilities are not commonly come across nowadays, okay, because nowadays we use more proprietary products, meaning the readily available commercial products to us. So all the commercial products would have actually undergone a lot of stringent stability tests whereby all the incompatibilities has been removed, okay, before they are marketed. So nowadays we don't see too much of incompatibilities during practice, okay. And most of the extemporaneous preparations that we do is also referring to the official formulas in pharmacopoeia, like BBC, okay, Martindale, literatures, okay. So usually these formulas again would have gone through stability tests and also long-term of stability data actually to support their effectiveness and also the stability of the drug throughout time. However, in practice, incompatibility may happen if, for example, we use the proprietary drugs, we need it, and we dilute the proprietary drug or we mix the proprietary drugs together and pack them or package them using inappropriate container or packaging. So then in that case, incompatibility can happen. So generally speaking, there are three broad categories of incompatibilities. The first category is what we call pharmaceutical incompatibility, and generally divided again into physical incompatibility, although there are maybe a lot more examples of physical incompatibility, but in this lecture I only provide, or I only summarize two common examples of physical incompatibilities, which are immiscibility and also insolubility. Another type of pharmaceutical incompatibility is what we know as chemical incompatibility. Again, there are a lot more examples of chemical incompatibilities, but I would like to explain a little bit more on terms of the pH and also cationic and ionic interactions. Therapeutic incompatibility is more commonly known as drug-drug interactions in the pharmacology lecture or pharmacotherapy lecture later on, and you will be exposed more later on. So drug-drug interactions, usually it can be divided into pharmacokinetic drug-drug interactions or pharmacodynamic interactions. So pharmacokinetic happens when a drug alters or changes the absorption, distribution, eliminations of a co-administered drug. Pharmacodynamic interactions occur when two drugs, when given together, they may cause additive effect, synergistic, or antagonistic effects. So in this lecture, I will not go into details in terms of therapeutic incompatibilities because these interactions will have covered in your PHD122 lecture previously, I believe. So the last type of incompatibility will be between the drugs and the containers, and we actually go through some incompatibility in the previous lectures on containers and packaging. For one well-known examples will be the softening of plastic containers by the methyl salicylate ointment. Okay, so in terms of physical incompatibility, basically it's an interaction between two or more substances that cause a change in terms of the color, odor, taste, viscosity, and morphology of the drug, meaning the shape, the structure, okay, the size of the drug. And this can actually cause non-uniform products being formed, and when a drug is not uniformly in terms of the uniform, it let its uniform lose its uniformity, then it's very difficult to get accurate drug dosage, meaning you will not be able to get a proper drug dosage if it is not a uniform product. Okay. So, however, the good things about the physical incompatibility is that usually it can be corrected by appropriate pharmaceutical skills. Okay, for example, let's see examples of immiscibility. Okay, so we know that oils and water are not miscible. They don't mix well together. Oils are immiscible with water. However, by adding emulsifiers, we can actually mix them well, okay, by the process called emulsification. All right. Another examples, for instance, if you want to add a concentrated alcoholic solutions of volatile oils. So this type of solutions, for example, spirits and concentrated waters, are usually flavoring agents. When you want to add this type of flavoring agents into an aqueous preparations, okay, so it may cause oil separations. So, but there are certain techniques that we can use to avoid or to reduce the risk of oil separation. For example, we can actually gradually dilute the alcoholic solution with the vehicle or the water, or we can actually pour the hydroalcoholic solution slowly into the vehicle with constant stirring. This can actually reduce the chances of oil separation. Another examples, for example, the potassium citrate mixture. Okay, so potassium citrate mixture is a preparations whereby it contains a high concentration of electrolytes. Okay, so when we want to add the the high concentrations of electrolyte to a aqueous solutions, which is saturated with volatile oil, it can actually again cause oil separations. All right. So, for example, if we want to add the volatile oil, the lemon oil, okay, the lemon oil into the potassium citrate mixture, it actually sort out the lemon oil. The oil separated out. They don't mix well together. So, in order to overcome this, we can actually add an emulsifying agent, which is a Kila tincture as an emulsifying agent. This will actually prevent the separations of the oil. Okay. Another examples will be relevant to insolubility. For example, some liquid preparations that contain indivisible solids. Sometimes we add thickening agents like tragacan, acacia gum, and we can actually able to form a uniform doses or uniform products. Okay, for example, chalk and sulfamidine are insoluble solids. So we can make it into a suspension by adding thickening agents so that these particles which are insoluble in vehicle, in solvent or water, so they will form as suspension. Another examples like calamine and zinc oxide, they are not soluble in water or vehicle. Okay, so usually they will be formed as something suspension, but it's as a lotion form. All right. Another examples will be, for example, some of the insoluble powders like sulfur, corticosteroids, some antibiotics, they are very difficult to be vetted by water. So we can actually add vetting agents to help to distribute the powders better. Okay. So examples of vetting agents like saponins and polysorbates. So having gone through the examples, the common examples of physical incompatibilities, we can actually understand or we actually find out that these incompatibilities can be overcome by additional steps or additional ingredients or excipients. We can add in additional things. So these incompatibilities can be overcome. So unlike physical incompatibility, chemical incompatibility is the reaction between two or more substances and it changes the properties of the formulations. And these changes can be in terms of precipitations, effervescence, degradation, color change, or and sometimes more extensive explosions. Okay, so chemical incompatibility, unlike the physical one, sometimes it's a bit to be difficult to be reversed. So before we add any agents, it's very important that we really examine clearly the chemical structures and the properties of drugs before we add them together or before we add them into a formulations. This is basically to avoid chemical incompatibilities. So the common examples of chemical incompatibility, for example, in terms of pH or dispersed system. So let's look at these examples. Most of the drugs are usually salts or weak acids or bases. So they are, when these salts, when they are available as salts, they are very soluble in water. But the unionized form of the acids or the base, they are not soluble. They are not water solubles. So take, for example, if a solution contains a weakly basic drug and you add something to the solutions, and this makes the solutions more alkaline. When it is more alkaline, the basic drug, the free base drug, basic drug will actually precipitate out. Okay. Likewise, the solutions of a weakly acidic drug, if we make it more acidic, then we will see that there's precipitations of free acid. Right. So another examples will be, for example, emulsions that made with soaps as the emulsifying agents. They are not compatible with mineral acids. If we add them together, then it will actually destroy the emulsion, and then the fatty acid will be precipitating out. Right. So cream also, if you want to mix cream, you need to make sure that the both cream are compatible with each other. So cream may come in different pH. Yeah? So the different pH actually, if it is not compatible, it may actually cause degradations. So another examples common of chemical incompatibility is about cationic and anionic interactions. So many of the organic compounds in the pharmaceutical formulation, it carry either a large cation or anion, meaning cation is like positive charges, the anion is negative charges. So if we add these two compounds with opposing charges or opposing ions, it actually can cause precipitations. Take for example, if we add chromomazine, which is a cationic drug, with phenylbarbitone sodium, which is an anionic drug, in a vial, for example, or in a infusion bottle. So you will start to see that the drug precipitate out because the cationic drug and anionic drug, they interact each other and then the drug precipitate out. Another examples will be, for example, the cationic medications or preservatives that comes in cationic, and if we add anionic suspending agents, these actually cause interactions with the ingredients in the suspensions, and they may actually impair the stability of the suspension. So with that, I would like to end this lecture with several references. If you would like to read more in details, or you would like to get more information about drug stability and also incompatibilities, you are recommended to read the Autumn's Pharmaceutics, which available in the library as older editions. So for older editions, you may look into the appropriate chapters from the book. Okay, it may be slightly earlier chapters or later chapters. Yeah, you may also look into the Remington Essential Pharmaceutics for chapter 4 or Pharmaceutics by Bharat on chapter 9. So with that, I end this lecture. Thank you for your attention.